Agriculture and Development Seminar Series. The SEARCA ADSS is meant to encourage the presentation and discussion of development and research issues, as well as their implications for agricultural and rural development and natural resources management. Thus, we encourage the audience to engage in the open forum after our speaker's presentation. We are very fortunate to have with us for our speaker this afternoon a research scientist with nearly 20 years of biotech experience in the discovery and development of biologics, natural products, and diagnostics. He has served in multiple senior management roles in R&D and business development with several early-stage biotech companies in Australia and Europe, including Patris Limited, HealthLinks Limited, formerly Krypton Pharmaceuticals, Serion Pharmaceuticals, and Morphosis. Vic also holds an honorary principal fellow or associate professor position at the University of Melbourne since 2007. Vic was an integrated academic program scholar, or INTAPS as we know it, at the University of the Philippines, Los Baños, so he is a homegrown talent. And as expected, he graduated magna cum laude with a degree in biology, major in genetics in 1985. His mentor was the renowned and multi-awarded research scientist, Professor Dolores Ramirez. He finished his PhD in microbiology, immunology from the University of Tennessee Center for the Health Sciences, together with his wife, the former Jocelyn Nunn. They migrated to Munich, Germany in September 1994 to work as a scientist with a startup biotech company, Morphosis. At Morphosis, Vic became part of the team that developed the only totally synthetic human antibody library, UCOM. Did I pronounce it? Yep, selected by Phage, is it? Phage display, and is used by majority of the top 20 pharmaceutical companies worldwide. In 1998, he founded Serion Pharmaceuticals AG in Munich, Germany, combining antibody phage, display, mass spectrometry, and Kali, chromophore-assisted laser inactivation. Maybe later you can explain it to us further. Technology as a functional genomics, proteomics platform. From Germany, Vic moved to Australia in 2004 to join Krypton Pharmaceuticals, a biotechnology company engaged in the discovery and development of protein-derived medicines targeting cancer, cardiovascular disease, and chronic inflammatory disorders. During this time, he was instrumental in the development of the ovarian cancer diagnostic Obiplex, which is now currently sold in parts of Europe and Asia. He also successfully led the team that developed a technology for isolating cryptic peptides, including one derived from milk protein that can reduce body weight and fat. That would be very interesting. In 2007, he joined Patris Limited to assist in its IPO, or initial public offering, at the Australian Stock Exchange, raising $25 million and established its recombinant antibody capabilities. This led to the successful development of a manufacturing cell line for their lead IgM candidate, which represented a major bottleneck for commercialization as well as the filling of new patent applications. Since 2010, Vic established Molawin Creek Ventures Proprietary Limited, his consulting outfit providing a variety of R&D, business development, and financing advice to early-stage companies. This includes Horizon Science Proprietary Limited, a private biotech company where he serves as Chief Scientific Officer since 2011 to help in the development of its sugarcane molasses extract as a functional food ingredient and dietary supplement for glucose and weight management. It is indeed an honor to us here at SEARCA to have Dr. Ilag here with us this afternoon to discuss about a common, chronic, and costly disease which according to the International Federation of Diabetes has affected 382 million people in 2013. And according to World Health Organization, it predicts that it will be the seventh leading cause of death in 2030. This seminar is very timely because I think in last November 14, we just celebrated the World Diabetes Day. So ladies and gentlemen, to present to us one of the solutions, perhaps one of the solutions for managing and curbing diabetes epidemic through his paper entitled The Biology of Diabetes and Its Agricultural Solutions, please welcome Dr. Leodevico L.
Okay, thank you for the kind introduction. So for this talk today, I thought I had to divide my talk into three parts. First, I think it's worthwhile to go through the basics of diabetes, and to have an understanding of how diabetes comes about as a disease. Then secondly, I think also to tackle the issue of understanding the mechanism of how diabetes occurs. Because still in active research, even though there are apparent treatments, one should realize that these are only ways of managing the disease rather than treating the disease. And then lastly is then to address what I have there as towards the end of question mark, a possible solution by agriculture. And I guess this was more something I had to tailor because I was invited, I think a couple of months ago, to give a presentation at the crop science cluster. It's only during that invitation that I realized that we could actually combine a potential solution with currently what I'm doing right now. So as a disclaimer, I'd like to point out I'm not an MD. So I do not intend to prescribe, or whatever I say here is not a prescription, and you should still consult your medical doctor. All right? Okay, so I think the first slide, I thought it would be worthwhile to go through diabetes basics. So there are two types of diabetes, as everybody knows. You have the type 1 diabetes, which is the juvenile diabetes. Oftentimes it's called early onset diabetes. So when you're young, you develop this disease, and oftentimes associated with people who are unable to produce insulin. So I know Gary Valenciano, so he's the typical poster child for type 1 diabetes. That only represents about 10 to 15% of the population. Now the most common one, which is rapidly rising, is type 2 diabetes. And in contrast to type 1 diabetes, there's called adult onset, and it's associated usually with lifestyle, having less exercise and eating the wrong diet. And this one, to differentiate from type 1 diabetes, is called insulin resistant. So in this situation, people produce insulin, but it's just that the person isn't able to respond to insulin and have diabetes. So there's that differentiation. However, it's been noted that by 2030, the Philippines would rank among top 10 countries which have diabetes. Actually, would be predicted to be number 9. I think it's going to be even faster than that, not 2030, but even earlier, just from, and I hope to convince you with studies which would indicate that that would be the case, mainly because of the size of the Filipinos we see today. All right, so the first thing probably to go through first is to understand the basics or the biochemistry of eating. So essentially diabetes, when you hear about diabetes, it's about blood sugar. So the question is, why is your blood sugar high? So I think it's worthwhile going through the process of eating, essentially how your body generates glucose. So when you go eat something, let's say a piece of bread, it goes through your stomach, gets digested. As you know, starch is the major component of most carbohydrates. The carbohydrates are broken down into subunits, mainly glucose, and it's only the glucose, the single moieties, carbohydrate moieties, which are transported to the bloodstream. Once it's in the bloodstream, it then gets absorbed by cells. There are receptors, glucose receptors of the cells, which then take in the glucose molecules, and then, since glucose is the basic energy of the cell, through glycolysis, and then you go through the Krebs cycle, as you remember your biochemistry, and then convert to CO2 and water. Right, so that's the basics. So what you can see here is before glucose gets transported, it has to be digested and transported to the bloodstream. I don't want to confuse you with this, but essentially what happens when you eat is food is taken in, broken down, glucose rises, and there's a complex series of reactions involving different hormones which regulate the processing of glucose. So insulin is the key molecule. Once you get insulin produced by the pancreas, it then tells your body, the muscle cells, the fat cells, with liver cells to make glucose receptors, so it would take in the glucose, so to reduce the amount of glucose in your blood. When you're sleeping at night, you don't produce, you could still produce glucose, but that's via the liver using glycogen, which is a hormone which breaks down, there's a hormone called glucagon which breaks down glycogen into some units of glucose, so you can use that. So overnight, you can still produce glucose even without eating. Now, when you go to the doctor then and have a evaluation of your blood glucose, or what you call here the FPS, fasting blood sugar, you have at least an 8 to 10 hour window where you don't eat anything. And then what you usually measure is your fasting glucose. If you're normal, there's a certain level, 110 milligrams per deciliter. At a certain threshold, from 110 to 125, you're either impaired for fasting glucose, that means you're a bit pre-diabetic, and about 126, you're then having diabetes. There are other parameters which you can measure, which is the amount of glycated hemoglobin. So this is a protein in your blood which has glucose molecules added to it. And you could also measure that. With the threshold, you should have about 6% of your hemoglobin molecules, less than 6% to be normal, and above that, you're either pre-diabetic or diabetic. Usually, in different countries, I think Philippines doesn't do that. We usually rely only on this. But one of the indicators of being pre-diabetic or early signs before you become diabetic is you're either impaired in fasting glucose or impaired of glucose tolerance. There's a test we could do where you take 75 grams of glucose and follow your glucose level, and it would tell you whether you're starting to develop insulin resistance. As I earlier mentioned, type 2 diabetes is insulin resistant. So if you fall around this number, about 110 over 125, it has been shown that 80%, you have an 80% chance of developing diabetes within 5 to 10 years. So the idea is you have to keep your blood glucose, actually the norm is to have this even below 100, not 110. WHO has had it as 110, but most countries usually have it as 100 as a cutoff. So this just shows you oral glucose tolerance test. So after overnight fasting, if you're normal, you have your fasting blood glucose here at 5.5. Of course, you're given glucose, then of course you have an increase in glucose, but that stimulates production of insulin, and then you have a rapid decline of glucose after 2 hours and go back to normal. So that's below your fasting glucose. If you have impaired glucose tolerance, there's already you start higher, but then you have a high peak of glucose, then it takes a while and it never goes down below your threshold. So that level is your impaired glucose tolerant or pre-diabetic. When you're diabetic, it's really high fasting glucose and then it really stays up. So the glucose taken in is almost not clear. That's why it stays longer in the bloodstream. Now, this is an important slide. So this actually traces the pathogenesis or natural history of type 2 diabetes. So if you go through this horizontal line, so you have here a lean, normal glucose tolerance, so blood glucose is normal. Then you become obese, you're still normal glucose, and then you become obese, impaired glucose tolerant. You start to increase your fasting blood sugar, and over time, you develop diabetes. But at that time, even though you think you're obese normal at this stage, what you don't really realize is that your insulin levels are already starting to pick up, and your relative efficiency of glucose uptake is decreasing. So if you're just starting to gain weight, even though you go to the doctor and you think your blood glucose is normal, it's not really a good sign yet until you start looking at your insulin levels. And that's what I was indicating earlier. You see the kids right nowadays. When I was in college, we were a bit slimmer. But nowadays, I think they're a bit on the heavy side. And it's not surprising to hear that there are already people developing diabetes here at 30 years old or 40 years old, which means even if they're in their 20s, they're developing it. They're already pre-diabetic 10 years earlier. So even though we don't test for glucose, fasting blood glucose at that age, it's already a bit too late to catch. And I think what's worthwhile is once you're obese, impaired glucose tolerant, so that means you're already pre-diabetic, you're a bit on the heavy side, your BMI body mass index is above the normal, you already reach the peak of hyperinsulin. So you're really producing a lot of insulin, but the glucose uptake is decreased efficiency-wise. So it's just like a machine. You're already stepping on the gas pedal, but you're not going any faster because the efficiency is really dropping. However, that's the stage of being obese diabetic with high insulin. The next phase is you're an obese diabetic with low insulin. What's happening there is your pancreas is already malfunctioning, so that it's not making any insulin at all. So it's totally shut down. So it's almost like you're a type 1 diabetic at this stage. Now the catch-22 here is, okay, you're type 1 diabetic, so just add more insulin. Well, the problem with that is already insulin resistant. So you might keep on adding insulin, you're actually complicating matters. So the way I look at it is, so insulin is similar to, we just make a comparison for antibiotics. So for antibiotic resistance, you have a certain resistance level, you just add more antibiotics and it will kill the cell. But it reaches a certain threshold wherein the microorganism couldn't take any more of that or could still be resistant to the antibiotics, then they become totally resistant. The same thing with insulin here. So there's a danger, you don't want to reach the stage wherein your beta cells within your pancreas is totally dysfunctional. And what's dangerous here, apart from that, is you notice that people with really advanced diabetes, they start to lose weight. And the reason is because they're not using glucose anymore. The body needs to get some energy, and what it does is now it starts breaking down protein. So you have muscle wasting, so that leads to then you turn on. Yeah. All right? So, in essence, for a while, there were essentially, so I was mentioning, essentially what they were, diabetes has always been about modulating or managing high glucose level or hyperglycemia. This could be essentially worked out through these different mechanisms. What we know now, after more studies, that there's a lot of different processes which lead to hyperglycemia. So it's not just taking in a lot of glucose, it's a combination of kidney malfunction, muscle uptake, livers, even decreased secretion of fat. So it's a really complex metabolic disease. And what most medications right now are essentially geared towards lowering glucose levels. And what I hope to walk through with you is oftentimes in medicine what we try to do is treat the symptoms, but not really treat the disease. So these are the current drugs in the market right now, which essentially target only 4 of the almost 8 different processes affecting hyperglycemia. And most of these drugs, again, depending what your profile is, sometimes it won't work for you. Metformin is up to the drugs. The other drugs, I think, have also been, like the DPP-4 inhibitors seem to show really potency, but they have some certain side effects. And some of them, like DPP-4 is a tumor suppressor. So if you inhibit it, you might even promote tumor growth. So there are some complications with that, but most of the focus has been essentially on modulating the amount of glucose in the blood, not really treating the cause of why you have high glucose level. So this just gives you, I'm not going to go through that, so these are the different types of drugs modulating, used for treating, managing diabetes. And this is just a slide to show you that the American Diabetes Association strategy has been really focused on managing glucose levels in the blood, which compromises the preservation of beta cells, so because it tries to produce more insulin, most of the strategy, so that kind of overworks your pancreas. Whereas the trend now is to this pathophysiological-based approach, where you try to preserve beta cell function and with no side effects of hypoglycemia or weight gain. So weight gain is often associated with insulin. So the more insulin you take in, it leads to weight gain. So this just summarizes what I earlier said. So effective type 2 diabetes treatment requires multiple drugs, and oftentimes the best way is not to just reduce glucose, but to have a better understanding of how we can manage the disease by looking at its origins. And of course, to start early in treating natural history of type 2. So don't wait till you're diabetic. If you're pre-diabetic, that's still manageable. I think there are ways of preventing that. All right, so I'll shift gears here a bit. As I earlier mentioned, this is the second part, so I think it's worthwhile to understand diabetes and the mechanisms of how diabetes comes about. So there are four points here. So hyperinsulinemia, high insulin levels, inflammation, the kind of bacteria in your gut actually seems to be playing a role in diabetes, and diet. And they seem to be all interconnected. So I'll start again, showing the slide again, just a reminder, hyperinsulinemia, what's in diabetes, looks like your glucose in the blood is correlated to the amount of insulin produced. But the efficiency is decreased over time with a high increase or the high levels of insulin produced in the body. So our report, if you have a lot of hypersecretion of insulin, it leads to hyperinsulinemia. It has been suggested that this has been the cause of obesity and diabetes. So producing more insulin by yourself. And oftentimes this is triggered by environmental factors or whatever diet or foods you're eating. Again, there are many studies correlating that by producing lots of insulin, it leads to insulin resistance. So again, similar to analogy to antibiotics. The more antibiotics you use, you develop antibiotic resistance. This parallels can be also seen with insulin. And actually, there are a lot of correlations as well that if you increase circulation of insulin within animals and even humans, it then eventually leads to the development of diabetes. There's a recent study last year which then showed using transgenic mice that really pins down the role of hyperinsulinemia towards obesity. So the current model then was high-fat diet leads to obesity, then you develop hyperinsulinemia. That has completely changed because when you have animals which produce less insulin, you can feed as much food as they can take and they don't gain any weight. However, if they have high insulin levels, they easily accumulate fat and gain weight. So that those studies have shown that. It hasn't been shown yet whether this obesity then leads to type 2 diabetes. So that's still an open question. Now, I earlier mentioned inflammation. So inflammation is actually good. When you get sick, you get a fever, you have swelling. Essentially what it does is your body's reacting to a foreign agent and tries to remove it. So if you have a cut in your finger, it starts to swell. The reason it's swelling is that it allows your immune cells to penetrate that vasculature and try to destroy or remove that foreign agent. So that swelling is due to inflammatory factors which allows these immune cells to go through. However, it's also been shown that it seems that among obese people, there is more inflammation. Not that they have a fever, but there's a low grade of inflammation. So the vasculature of obese people seems to allow the penetration of more pro-inflammatory cells like macrophages and other inflammatory cells. Whereas the normal being, the vasculature, it could look like your blood cells, which prevents the fluids from going, not the blood cells, but your blood vessels, prevents fluids from penetrating or escaping your body or getting whole. It then prevents any of the cells from penetrating. So if you have an infection, these cells actually play a role in trying to clean up the foreign bodies or any other microorganisms or infectious agents. And this leakage of the vasculature allows these pro-inflammatory cells, which then actually stimulate your fat cells to produce more of these growth factors, which then trigger obesity. Actually, there was a paper out, I just read this morning, that leptin actually seems to be a promoter of breast cancer stem cell growth. So there's even discussion that actually leptin might be a target to develop a drug against. All right, so this one just shows you, let's go through this with how inflammation can be triggered by eating. So when you, from the gastrointestinal tract, you produce glucose and free fatty acids. Free fatty acids then are taken in by adipocytes. Now, adipocytes are your fat cells before they become fat cells. So they could differentiate into fully develop fat cells upon stimulation of free fatty acids. Once they get activated, they differentiate. They then send signals actually, so that these macrophages come in. And the reason is because there are some particles from the food after eating. Some of the fat cells lyse, and they have to be cleaned up. So it's a natural process that these pro-inflammatory cells have to enter the body or adipocytes to clean up those material. However, if there is constant stimulation, then you have a chronic inflammation. So most of your adipocytes will continuously differentiate. And actually, one of the triggers to differentiate adipocytes is actually insulin. So if you keep on pumping insulin, insulin then will cause differentiation of adipocytes, then you have more fat cells accumulating fat droplets. And this then creates a vicious cycle. Once these lyses then can go through this, you stimulate, and the more macrophages entering the neighboring area of your beta cells, then it shuts down the production of insulin. And so that's the current model of how this works. So just summarize essentially what I said regards to strong evidence of inflammation in regulating type 2 diabetes. Okay. So the other factor which I mentioned earlier is the role of the microbiome. So it's now becoming clear. There's a lot of correlative studies that if you look at the microbiome, the kind of bacteria in a diabetic or obese patient, you can differentiate it from the ones of a normal or lean subject. So it seems that although those are correlative, correlation does not mean causation. You have to show a direct causative effect. And one of the models right now is, again, in terms of gut permeability, one hypothesis is that the gut stomach tends to be permeable. And one of the triggers for inflammation actually is the translocation of bacteria into your system, which leads to metabolic endotoxemia. So once it passes through your system, it then is able to influence the liver, fat cells, and muscles. If there's inflammation in these different organs, it then leads to diabetes or other insulin resistance for muscle cells. So just inflammation within these areas around these organs leads to insulin resistance. So these are the key organs for modulating glucose metabolism. There was a paper a couple of months ago, or 3 or 4 months ago in Science, where what they did was, and this was more a direct causative demonstration of the role of microbiome. So they took 2 discordant twins, so one fat twin and a lean twin. They did a fecal transplantation, essentially they transferred bacteria from the obese and lean twins to corresponding germ-free mice. Then they were fed low-fat, high-fiber diet, and those mice which got bacteria from the obese twin became obese, and those from the lean twin became lean or stayed lean. Now the experiment even became more interesting when they cohabited lean mice and obese mice, because they could have horizontal transfer, the infection by being, their poop, and that's a way of transferring it. And if you have an obese mouse, even though it's low-fat, high-fiber diet, it cannot influence the phenotype of the corresponding lean mouse. However, the lean mouse could influence the phenotype of the obese mouse and try to develop a hybrid, so more reduced weight. And if you look at the bacteria of these intermediate-sized mice, they actually contain some bacteria of the lean mouse. And vice versa, if you have a high-fat, low-fiber diet with a lean mouse, and then it could be converted to a fat mouse by transfer of the fecal. So that demonstrates that the kind of bacteria present in the stomach influences the development of obesity and potentially diabetes. So this then slide shows you that fecal transplantation and the correlation as well as causation and the role of the gut microbiome in diabetes. So are there any other options then apart from medication? So this now goes to the food part. So have you heard of the glycemic index, GI? Alright, so this is popular in Australia. I think the recognition rate of GI is about 85% of the Australian population. So essentially GI is glycemic index. It measures the rate the food delivers glucose into the blood system. So if you look at carbohydrate-rich foods, this only applies to carbohydrate foods. Essentially what you're doing is, once you eat a certain kind of food, you can measure its glycemic index, and the index is setting glucose as 100%, or a glycemic index of 100. So if high GI, you have a spike in glucose, as I earlier pointed out, then it goes down rapidly, because once you make a lot of insulin insulin clears up the glucose, so that's high GI. Low GI is the peak. The peak is lower, but it stays longer in the blood because the insulin produces less and then it stays longer, this longer lasting energy. So if you're an athlete, you prefer taking low GI foods because you don't want to have a glucose crash. If it's high GI, your system will crash right away and then you would need more energy. You want something where it moves to the blood slowly. So this slide shows you the categories. If you're high GI, again, it's relative to glucose. Greater than 70 is high GI, medium GI is between 55 and 69, and low GI is anything below 55. So these are the foods which are high GI and low GI. So when you say about rice being bad for diabetics, to some extent it's true. White rice. But that's if it's jasmine rice, the one used in a restaurant. I think the sticky rice, I think is even worse, probably about the same. So you have chips, biscuits, potatoes, that's 100. However, there are the low GI foods here. So basmati rice actually low GI. So if you want rice and eat rice, I recommend you eat basmati rice. Don't eat jaguna. So you can still eat white rice. And then vegetables, of course, pasta. Okay. So these are the high GI versus low GI foods. Again, this shows you when they say carbs. So it's not all carbs were created equal. You have the good carbs and the bad carbs. So the good carbs are those which give you low GI. There's a spike, really low GI, because you produce less insulin. So less insulin then takes a longer time for your glucose to clear your blood. Whereas if it's bad carbs, are those which spikes your insulin and then leaves glucose crash. Once it's glucose crash, then you feel a bit weak, then you want to take it more. To keep the spike, and then as earlier showed, hyperinsulinemia, you keep on producing insulin, that leads to potentially diabetes. And so that's why it's not good to eat high GI foods. It's been, again, as Mark pointed out, so in Australia, this is recommended to eat low GI foods into the World Health Organization. So I think for the Philippines, something probably to worth, something to look at. Again, the evidence is there, preponderance of evidence is there, and it has been shown. There are many studies that actually having a low GI diet risks to develop, prevents or reduces the risk of developing type 2 diabetes as well as cardiovascular disease. There's a paper back, I think about 2003, did a meta-analysis of low GI diets, and actually it shows that by taking a low GI diet, it's almost like it's just as effective as taking anti-diabetic drugs. If you want to maintain weight, the best is to have a high protein, low GI diet. That's the best way. Because one of the problems after losing weight is the rebound effect. And it's better to have a low GI diet to be able to maintain your weight. So in Australia, they have a low GI symbol. So foods, when you go to the supermarket, have branded with this particular symbol. Essentially, you go to the Glycemic Index Foundation, you can then have your food tested, and if it follows the scientific standard, measuring the food, then they'll put a stamp. So when you go to the grocery store, you just have to look for the low GI symbol. It makes it easier. You don't have to do this calorie counting, like 2,500 calories, 100, no need for that. Just look at the GI symbol. Now, let's come to the next part. So is there an agricultural solution? So I've gone through the diabetes part, mechanism of action, how it comes about, the role of food. So of course, agriculture then combined to food. Now, the way we'd like to address this is go through some interesting developments, I think, in the field. So in 2004, David Sinclair and his colleagues at Harvard Medical School came up with a xeno-hormetic hypothesis. Have you heard of the xeno-hormetic hypothesis? So hormesis essentially is when an organism is under stress, it produces secondary metabolites for its survival. So xeno, because it means foreign, is a particular organism produces a metabolite which could influence that of another organism's metabolic systems for xenohormesis. So from this hypothesis, David Sinclair proposed that when the environment, let's say when the host, let's say plants, anticipate certain changes in the environment, let's say climate change, it starts to make some adaptations, not for only its survival, but also producing certain chemicals, phytochemicals, which would be useful for its consumers. So it's more an altruistic effect. So it's not the survival of the species itself, but survival of the whole ecosystem. And that, one of the supports for that hypothesis is if you look at the different, one of the popular phytochemicals are polyphenols. So plants under stress produce certain polyphenols, such as resveratrol, which is found in red wine. And these, like resveratrol, would affect different processes from inflammation as well as glucose metabolism, stress responses. And they have a positive effect. As you know, with red wine, when you make the grapes to make a good wine, you actually grow them under stress. By growing them under stress, they produce these better phytochemicals, and those phytochemicals are the ones which have a positive effect. So same thing with green tea, EGCG, and then curcumin, which is found in turmeric. So with that idea then, so not only do plants produce polyphenols or other phytochemicals for their own benefit, they're also producing it for the benefit of their consumers. Right? So that is the xenohermetic hypothesis. It turns out, actually, if you look then at the different modulating points of modulating diabetes, different processes from glucose absorption from the intestine, protection of beta cells, as well as suppression of glucose from the liver, as well as uptake of glucose by muscle cells. There are a variety of sources. Actually, these are polyphenols from these different sources. I only copied a few of them. There are several, which act on different parts of the system. So you have here black rice because it inhibits, instead of glucobase, I think there's a drug, acarose. You don't need to take glucobase. So when you eat rice, you might want to eat rice with beans or with other inhibitors which block the enzyme which digest the carbohydrates. So these are potentially potential sources. So there's ampalaya, if you're asking it. So ampalaya, turns out bitter melon or ampalaya contains polyphenols which increase the uptake of glucose in the blood by the muscles. So that seems the mechanism of action. All right. However, most of these studies have been done in vitro in animals, so there hasn't really been a lot of human studies, clinical studies. But that's starting to change. So as you know, cinnamon has been effective, and there are publications now showing that cinnamon extract can actually improve fasting blood glucose. So that by taking cinnamon, you can actually lower your blood glucose. So it actually might not be a bad idea to just go buy cinnamon. Would be a good one. Now, how much cinnamon they put in cinnamon, it's something worth. All right, so potatoes, I told you earlier, was a high GI. In Australia, actually developed a natural low GI potato by breeding purposes. So it's the charisma potato. So this is studies. The charisma potato is low GI, GI about 53. Most potatoes are in the 90 to 100. My favorite potato actually is the Nicola. The Nicola is medium GI, but that's why for diabetics, potatoes are not recommended. So this has been validated, this particular potato, the charisma potato, and I've tasted this. So you go to Australia, these potatoes are so hot, they disappear right away. So every week they sell it, and in terms of taste, it tastes exactly like any regular potato. And what it turns out is the structure of the carbohydrates of the potatoes are a bit more resistant to starch for alpha amylase digestion. So for us, now this is a plug. So I work with a company, Horizon Science. So we developed actually an extract from sugarcane molasses through a process of filtration. And we could extract the polyphenols, which we call benicarb. And if you combine benicarb with regular sugar, you reduce the GI. If you just use molasses with raw sugar, the GI is about 65. That's the rough estimate of GI. And white sugar is about 65. So sugar actually is a low GI. No, not low GI, it's medium GI, depending on the region. So if you talk about rice and sugar, actually rice, depending what kind of rice. I know jasmine rice is a GI about 80. So sugar, even though people get so fixated that, oh, I'm not going to take any sugar, but the amount of sugar you take in actually relatively less than when you eat rice. So I think they're So I showed you this slide. So this one is logic game. So we created this sugar together with Sugar Australia, and it's actually sold in Australia. So this is sold since 2009. So the sugar is sweeter, or it's just as sweet as regular sugar. Cocoa sugar from coconut is actually low GI sugar, but it's expensive. And compared to price, so this, considering this is made in Australia, is still 2 to 3 times cheaper than cocoa sugar I buy from, let's say, Robinson's supermarket. So this is low GI sugar. It's certified by the GI Foundation, so we get the seal that it's low GI. This extract, you could also then combine with different carbohydrate matrices, and in a dose-dependent manner, depending on the percentage, you could have a corresponding GI percent reduction. Even white bread, so one of the things we'd like to do is make a low GI pandesal. And it's actually, we're doing something here with a Cello restaurant, and I think, this is a free plug-in. I'm not going to get anything from it, but they are testing it. Turns out if you mix Benicarb, this molasses extract, with bread, it actually makes it more moist. They did a test when I was here about a couple of months ago when we started the testing, and they gave us a loaf of bread which they tested. We made a mistake of keeping it in the fridge, and after 3 to 4 days, my mother and I forgot about it, and we took it out, and actually the moisture was still the same. Ironically, well, not ironically, incidentally, then we're worried was the higher water activity. We have higher moisture would correspond to higher water activity contamination. Turns out it wasn't contaminated. We've done some tests then. And the reason is because polyphenols also actually have antimicrobial activity. In Europe, the trend actually now is to change preservatives from more natural sources, and polyphenol seems to be the substitute for making preserved foods in Europe. Maybe that's a trend in Europe. All right, so there are other molasses extracts you could look at. We've done some studies, especially in mice. You can take these other extracts and actually you can have more lean mice where they develop better lean mass, so just more muscle and less fat without changing the amount of food they eat. So they can eat as much as they can, and they're still not gaining fat, and actually they lose fat and gain muscle. Whether that translates to humans remains to be seen, and we're exploring that possibility. In my previous slide with cryptome, we also isolated a peptide. Again, it's not limited to plants. We can also look at different animal waste streams. So in this case, we've isolated a peptide from waste, from whey. And one of the things I realized, so in Australia, what they do is they collect whey and they process whey and extract the different components of whey. You could actually sell whey protein, and that's what's used by bodybuilders. In the Philippines, you know what they do with whey? Let's say a Carabao Center, and they throw it. Right? So it's not being utilized in the Philippines at the moment. All right, so this one then goes along the lines of what I was talking about with polyphenols. One of the things they're doing in Australia, in New Zealand, this paper is because polyphenols, as I earlier mentioned, plays a role in disease resistance. So what this group in Australia, in New Zealand is doing is, because polyphenols are have a health benefit, they're breeding apples based on the increased polyphenol content, because different apples skins have different polyphenol content. So they're using those polyphenols as quantitative trait loci to breed different apples which have multi-polyphenol content. Now, from a plant breeding point of view, I know we have to make transgenics, but it might not be necessary. It might be worthwhile exploring. You can hit 2 birds with 1 stone. With the health benefits of polyphenols and other metabolites, if you could use that as in your breeding program to isolate or develop disease-resistant plants, you could also at the same time have a reservoir of different or high-value products, such as polyphenols, which could have other uses. So this essentially summarizes my top 10. Current paradigm is mainly looking at glucose levels. I think there should be an effort more towards managing the disease by, let's say, looking at promoting growth of good bacteria, and reducing inflammation. failed to mention earlier, so polyphenols actually turns out to be a good prebiotic. It helps in the growth of healthy bacteria. And then there are new opportunities for extracting polyphenols or finding chemicals from animal metabolites, so that hasn't been explored yet. And one of the things I found attractive, so for the past 20 years we've been working with drug discovery, developing antibodies, and these are expensive things to do. And in drug discovery also, you try to go to these marine products, you go find these conch snails, and go for really exotic or esoteric organisms. But what's probably interesting is what is overlooked. And oftentimes the drug fails is because you cannot make it. It's so expensive to make. But I think just realize when doing my stint here, people have overlooked the potential actually of agricultural waste. So I showed you something here about molasses. Molasses is cheap. So we found an ingredient there which would lower glycemic index and possibly for weight management. But I think it hasn't really been pursued yet and explored the possibility of look at these industrial wastes as a source of new compounds for pharmaceutical purposes. So the term farming, P-H-A-R-M-I-N-G, was used before when they were trying to use animals, farm animals, to make transgenic human proteins. So they use farm animals to make transgenic protein. But rather than doing that too high-tech approach, but rather look at farm as actually farm products as sources of bioactives. And then new opportunities for developing plant strategies based on xenohormesis. And again, lastly, opportunities looking at dairy waste, source of xenohormetics. And just because of this balance of xeno or ecohormesis, where the interaction of the plants and animals could be looked at and try to tap the different responses could be source of new compounds for health benefits. And they probably had this slide then and talked with us. Not ironically, but Hippocrates, the father of medicine, actually said that let thy food be thy medicine, and let thy medicine be thy food. So thank you for your attention.
Thank you very much, Dr. Ilag, for that very informative discussion about diabetes. May I now open the floor for questions from the audience? Yes, ma'am. Kindly introduce yourself and the organization you are connected with.
Pam Fernandez from Crop Science Cluster UPLB. I'm intrigued by the phenomenon of xenohormesis because it means that the plant actually exists in the service of the earth and mankind. Isn't it what it means at the greater or grander scale?
What was the question?
The xenohormesis, isn't it that, you're saying that it produces something not only for itself, to help itself, but for the welfare of the consumer or the animals, the plant, the environment, humans. So it means that they are not just for themselves, but really to nourish the earth in general.
Yes, I think, my feeling, I think, from David's postulation, I think it makes sense also from a point of evolution. So if you think of evolution, of course, evolution acts on a population. But through this process of evolution, the cycle, again, depends what your view on climate change is. You could be working in different cycles. So whichever gets selected for are those which are able to then work together. And just like evolution is a constantly working process, there would be constant selection and, of course, extinction of other organisms which are not able to adapt. And I think with that in mind, we could then utilize that adaptive process or evolutionary abilities of, again, that's why I kind of extended it, not just xenohormesis of plants, that's why animals as well, because other animals, like cows eat plants, cows have other wastes. And it turns out actually cows are interesting because if you look at milk, Milk have a lot of proteins that haven't been explored yet. One of the popular ones is lactoferrin, which is compared with, which is really has some multifunctional property. But lactoferrin can be broken down into different pieces, which we call cryptines. And each piece of lactoferrin, different proteins within that milk protein, would have specific effects. So that's one of the things. I didn't go into detail, but we've isolated a fraction which could lower fat metabolism as well as glucose metabolism. So that interrelationship, I think, is worth exploring. So instead of just reading logically, it might be worth looking at what nature has done for the past billions of years as an alternative strategy. So that's why I thought from the agricultural point of view, the obvious thing is, oh yeah, we just make it transgenic. But probably might be worthwhile as well. What has been nature doing for this billions of years? And to tap that. And I think one of the things I think there might be an attractive, what do you call that, proposition for agriculture. Because agriculture has always been a commodity business. On the pharma end, it always has been on high value products. The trend right now, if you look at these major food companies, is they're trying to meet halfway. There are advantages of food that it has safe, but if you make functional food or medical food, it could be able to address a high value market and move away from a commodity market. But again, one of the limitations, I think, especially with agriculture, because the focus is always on production, making more. But if you can produce and show that you could have a bioactive and do the right type of trials, which I think that's what's missing as well. Like, tulad ng maraming yung malunggay. So malunggay has been shown as some health benefits, but there's really been no controlled studies in malunggay. So people then now start formulating, making dry forms, making pills, capsules, but we don't know whether that is the effective dose. And then people, my thinking is, the placebo effect here is very high. So even though probably it's not an effect of a compound, so until we probably do more controlled clinical trials, and that's probably something agriculture could learn from the farm end, so that there's validation. Because once you validate that, then you could have a really strong basis for them saying that, yeah, this is active. And I think that's been probably the Ampalaya type of thing, Ampalaya, that you kind of just cook everything from Ampalaya, but not thinking that, well, what kind of form is really active? Because once you start cooking, that's where chemistry comes in. You don't know whether you destroy the active compound. It's like one of the polyphenols, which is popular, is quercetin. A rich source of quercetin is onions. However, if you cook onions, 90% is removed. So it's better to eat onions raw. So when I usually eat a Big Mac, my wife complains to me, but I say, well, I have the onions here. So because quercetin is one of the inhibitors also of glucose transport, as well as inhibitors of alpha-amylase. So there's some justification there. If you want to eat a Big Mac, yeah, it's okay. Once you have those onions. Don't eat too much though.
So Dr. Ilag, what you're trying to say to us is, let us not cook our food, eat it raw.
Might be helpful. Unless you go really but the molasses, like we're studying molasses. If you look at molasses, that has gone through a lot of high heat, high pressure. That's what we're attracted to it. Molasses, naturally, I'm not giving you a price. So we produce tons of that just by filtration. We have 2 people who just produce that. We have a plant in Osman, far north of Queensland. And from a ton of molasses, the value of the product we get is much more than ethanol. But we're still producing ethanol. It's just a teaser. And if you look at the analysis of molasses extracts, the polyphenol, it's really complex. So it's really exciting as scientists that this is like a gold mine. And because you produce tons of this material, you don't have problems. So I'm talking with other people, so we just have to think of different wastes here. One thing is probably like buko, tubig ng buko. Actually, when you take, I was talking with Tony Laurena, so if you take this coconut water, fresh coconut water, you put it on your white clothes, overnight it turns yellow. That means there's polyphenols there. So have we looked at that? No, just keep the chopping and just throw it away. So there might be some things there. So just think of other ways. Rice bran is another interesting thing. So rice bran oil is sold in Australia. So rice bran oil actually turns out to be really good. So we use rice bran oil. The reason is rice bran oil has a high smoking point. And that's what's used by Japanese restaurants. So when they make their tempura, you have to always boil it. Olive oil has a low smoking point. That's why it's not good for frying olive oil. But rice bran oil is what you use, and it doesn't have any taste. And rice bran oil, okay, but are we making rice bran oil? No. And the price of rice bran oil in Australia, that's the one we use, is much cheaper than olive oil. So that's the frying oil we use, and actually vegetable, a salad oil as well, because it doesn't have a taste and smell. And it's rich in oryzanol, which is a higher boron value than the oil we like. So just thinking, so you don't really have to then go to these esoteric organisms, try to grow them, and then you're stuck. You have an active compound, how much can you produce? A microgram? That can go anywhere. Even though you say it's active, yeah, you have a nice publication. But we're sitting on lots of agricultural waste, and that has lots of things. And again, along the Xenohermesis hypothesis, if nature, and then especially with climate change, is already adapting, that means the plants are already helping us with something. So instead of us panicking, oh yeah, what we're going to do, we've got something, actually we might start focusing on solutions which are already being worked on by our producers for COVID happens.
Yes, Dr. Marcy.
I'm Chief Gordon Steele, and I'm a nutritionist with the Institute of Human Nutrition and Food. And my thoughts are on the glycemic index. Listening to you, sir, it's it's a concept so easy to apply. But I would like to share my experience in the glycemic index. It's so difficult. It's a very difficult concept to understand and apply, at least in the Philippines, because as we all know, the kind of diets that we have are mixed. They come in mixed food combinations. And to my knowledge, GI is not something like mathematically 1 + 1. It's a food with a GI, with another food with another GI. The result is could go higher or could go down. So with that, how do we apply the GI considering the diets, the typical diets that we have? Also, is there any update also about the GI and the glycemic load? Because I understand that the glycemic load might be a better concept to apply in terms of, say, diet counseling, which I am into. Thank you.
Good question. So glycemic load. So glycemic index is just the index of how fast a particular amount of food enters your bloodstream. But of course, even though it's low GI, let's say I have a low GI food there, but if I eat 1 kilo of that amount, it's going to be a lot. So that's what you meant by glycemic load. Now, if you have a high GI, but you only eat too little of it, then that kind of, you might still be off. So the question is on the quantity. But in reality, I think it's difficult to really estimate totally how much you've eaten. So the way I think control one of the things when you can find in general, I think you can only estimate in terms of low GI or high total glycemic load. But I think what's the advantage of having a low GI is because the amount of, you tend to be satisfied faster because of the amount of glucose being present in your blood. There's no hunger or glucose crash that you need to search for more food to be taken in. So just by having conceptually, if I eat this X amount of food that I know is low GI, that should give you at least a good approximation. Because even with the calorie counting, the problem with that is really,
how exact really is the calorie count, and you really don't know. And then calories, I think, in my opinion, really don't really add up. I don't know whether you heard the recent study using sucralose. So sucralose is aspartame, not aspartame. Sucralose is Splenda. They did a study. So Splenda is 0 calorie. So this study was probably about 3 months ago. What they did was they did an OGTT test, oral glucose tolerance test. So what they did was they take Splenda first, population, took Splenda, I think 15 subjects, and then took the glucose, 75 grams of glucose. Those who did not take Splenda and a normal glucose tolerant, glucose peak, those which took Splenda had a 20% higher insulin and glucose peak. That's 0 calories. So calories doesn't tell you everything. And that becomes worse because the theory then now is, although they're blaming increase in sugary drinks, but nobody has really looked at the amount of the correlation with those taking in diet soft drinks. Because there's an emerging trend now that diet, low calorie, 0 calorie. So if you translate that study, then what that would suggest is if you take a Coke Zero and eat a Big Mac or a quarter pounder, your spike in insulin would be higher drinking a Diet Coke, which is 0 calories. So the GI of that, essentially, by taking sucralose, you've actually increased the GI. I'm not sure I answered your question, but I know it's difficult, but I think at least gives you because nobody really follows it, but at least you could have a shopping list that people know that's low GI, I eat that, certainly, and I'm conscious, and I think the total effect would still be better than that. But of course, quantity has to be watched. Yeah.
Any more question? Oh yes, Dr. Bray.
So I would like, I'm Nuevo from Crop Science Cluster, and I would like to know what or how would that molasses be of importance to diabetics?
Yeah. So, you mean the molasses extract you're working on? Yeah, so we're doing clinical, we're still planning to do the clinical trials. So we've done, okay, there was a, I could talk about. What we've shown, if you take that extract by itself before you eat, your glucose response also goes down. So, and what's more interesting is the insulin response is also lower. So you produce less insulin when you eat. So in terms of diabetics, it lowers the GI, let's say your carbohydrate intake, because you still need carbohydrates. I think people get this misconception that, oh, you shouldn't have any sugar at all. I mean, you still need sugar because that's what drives or helps, that's what fuels you. But by lowering the glycemic index or regulating the amount of glucose going through your blood, the rate of amount of glucose entering your blood, then it would help you manage your diabetes. Did that answer your question at that point?
Yes, but I'm just wondering whether what would be the volume of intake for a diabetic for it to be, lower the glycemic index? Any volume of the...
Nothing really. I think depends what because molasses is considered generally regarded as safe. Based from what we've done, and at least the European Food Safety Authority also had the same opinion, like what we produce is similar to molasses, even though we filtered it only for the lower molecular weight components. So, in essence, you could still take it. You won't be able to treat your diabetes, but it will help manage the amount of intake. So instead of, let's say, taking rice, 80, now we haven't tried adding this thing with rice, but if you could lower your intake, you don't have this glucose spikes, which would help manage your diabetes. So that's why low GI foods are recommended for diabetics, and even for pre-diabetics.
I think that's still up.
Question number 1 is about substituting potato with sweet potato. And then number 2 is how do you test your, when you do your sampling, because you know that the liver has different functionalities or operation during the day and in the evening. Like for example, for sugar, carbohydrate metabolism, it's more active, able to metabolize it from 3 o'clock in the evening up to, the afternoon up to morning, 3 o'clock, diurnal pattern. And number 3 is coconut, because coconut water is said to be not good when you take it in the evening.
Oh, wait, that one I don't know.
And I do notice that. I don't like coconut in the evening. It gives me like a sharp pain. And I don't know if it's psychological, but I've heard it from the old people that you should not anymore drink coconut water at night.
So, now the third question I cannot answer. I don't have an answer. But the first one I think is a good point. So sweet potato, actually if you go to the grocery, so I live in Australia now, they have it super fries. And actually what it is cut sweet potato. And so if you want to substitute sweet potato for potato, possibly, but I think that it's still different, tastes different. But if you want as a staple, I would say actually sweet potato is better than this golden rice. No offense. I would take that. I don't have anything against transgenics. I'll still eat GMOs. But if you're really for practical purposes, because I think the nice thing about sweet potato, it's rich in fiber. That's one of the things which could lower the glycemic. And it's a low GI food, rich in fiber, and you have this carotenoids, so vitamin A. So the question is why you would develop golden rice. Of course, the question then for golden rice, I don't know much detail, is it high GI? Because then you have the risk. You have a high GI rice is high in, and it could pose some problems as well. It's vitamin A, you have retinoic acid, it could stimulate growth. But it's not nature that kind of involvement. The liver, I couldn't comment. I don't know the answer.
Okay, so that wraps up our open forum. If you need to take a closer look at Dr. Ilag's presentation, you may download his PowerPoint from the SEARCA website. And I failed to mention earlier that Dr. Ilag is a UP visiting professor, and he's based here now in Los Baños because he is being hosted by the UP System Philippine Genome Society. So he'll be around, I think, until the middle of January next year. So, yeah. So let's give him a big round of applause.